the experiment that we are going to demonstrate is starch test for the given food samples. The starch test is relevant for grade 10 life process chapter, where we have a topic called photosynthesis. We all know the equation for photosynthesis, that is, we need carbon dioxide and water. In the presence of sunlight and chlorophyll, the plant will synthesize glucose and oxygen. Oxygen will be liberated by the plant body to the atmosphere, whereas glucose is an energy-rich compound which has to be stored in the plant's body. Plant will not store glucose as it is. It gets converted into a complex form, that is starch. So the plant body will store food in the form of starch. So today's experiment's aim is to detect starch in the given food samples. Principle behind this experiment is that starch in the presence of iodine solution produces a dark blue coloration, which is very evident. This is because iodine gets trapped in the helical structure of starch. So food items which have starch will show positive test for the experiment. The materials required are petri dish, potato, table salt, sugar cubes, boiled rice, iodine solution and dropper. So let's begin the experiment. We have set up all the given materials in the order. So as you can see, I have arranged all the necessary things. We have taken boiled rice, potato, salt crystals and sugar cubes. We have iodine solution with us, and to help drop it onto the given food items, we have a dropper. So first we will test it with our normal food items like salt and sugar. So I'm taking few drops of iodine solution and dropping it on our sugar cubes. So as you can see, the iodine solution is kind of orangish yellow and sugar cube hasn't changed any coloration. Now coming to our salt crystals. So if I drop iodine solution onto the salt crystals, the coloration is as it is. Now we will go to potato sample. So potato, I've taken raw potato, I've cut it into half. I'll just drop few drops of iodine onto the potato. So you can see there is a bit of dark stain which is being formed on the potato. Now at last we will take it to the boiled rice. Now boiled rice, as soon as I drop, as you can see, it is turning very evident bluish color, correct? Whereas potato on the other hand is taking little bit time because this is raw potato. If we had taken a boiled potato, the result would have been even more evident. So we can see that potato and boiled rice have taken the stain, whereas sugar and salt crystals have not taken it. So from this experimental procedure, what is our observation? Observation was that only food which contains starch will show positive test with iodine, that is our potato and boiled rice. Whereas other items like sugar cubes and salt crystals did not show any positive result. What is the inference that we get from it? We know that carbohydrates are energy-rich compounds, commonly known as sugars. They are divided into simple sugars and complex sugars. Simple sugars are present in sugar cubes, examples, glucose, sucrose, etc. Whereas complex sugar is present in potato, that is starch. So simple sugars have very simple structure. They have C6H12O6 glucose or sucrose. Whereas complex sugar are formed from simple sugars. Like glucose will bind with another glucose molecule, and like this they form a chain and thus form a helical structure, as you can see here. Now once this helical structure is formed, now as soon as we put iodine solution to them, the iodide ion gets trapped in the starch helix. So the test will become positive to only food items which have the starch present in them. Thank you.